This meeting is now being recorded.
Thank you for joining us for Metabolon's half-hour webinar series, where our topic today is Precision Metabolomics, a single technology for understanding human health. We'll have a short period of time for Q&A at the end of this webinar. Please feel free to submit your question via the Q&A section at the upper right of your screen. And just a quick reminder, this webinar is being recorded. Thank you. The presenter for today is Dr. Annie Evans. Dr. Evans leads the Global Metabolomics and Lipidomics Profiling Research and Development Team at Metabolon, where she has been working for 13 years. The metabolomics and lipidomics platforms developed under Dr. Evans have been the analytical basis for thousands of commercial studies from over 500 clients since 2004. She has publications covering the analytical methodology, as well as informatics and data processing requirements for global profiling workflows. Dr. Evans received her PhD in chemistry from the University of Virginia, where her research was focused on biological mass spectrometric detection of protein and peptides with renowned professor Dr. Donald Hunt. Currently, much of her research is focused on developing the global metabolomics and lipidomics technologies, as well as new technologies to be able to support initiatives into large human cohort studies to study human health, while also making the technology precise and robust enough to study individual patient health, precision medicine. I'll be turning the presentation over to Dr. Evans now.
Good morning, and I'd like to thank everybody for taking the time this morning, if you're on the East Coast, to tune into this talk. So as introduced, I'm going to be talking today about precision metabolomics, and while I promise the rest of my talk is not going to be like this, I actually would just love to read this paragraph to you, which was a paragraph pulled together by one of my colleagues, and I think it does a fantastic job summing up what precision metabolomics is. So precision metabolomics is an approach to metabolomics that is capable of measuring metabolites with sufficient accuracy and precision such that N of 1 studies of an individual is possible. This approach uses extensively controlled processes to provide data with the smallest degree of variability in the measurements, usually less than 5%, allowing accurate and precise identification of the maximum number of biologically relevant metabolites in a sample. And the goal is to comprehensively and reproducibly map metabolism and metabolic changes induced by perturbation of disease and treatment with sufficient precision to produce actionable information at the level of the individual. So that, in a nutshell, is what precision metabolomics is. It is essentially an approach to metabolomics which takes the data to a level where we can start to use this technology in studying individual human health. So why do we even study small molecules or metabolomics? So one of the nice things about small molecules is that not only are you getting the natural processes that are the endpoint of genomic, the hierarchy of biology, in that genes are transcribed into RNA, which are translated into proteins, and then proteins carry about their business within the biological system using reactions and products in the form of these small molecules. So studying the small molecules in these samples, you can see what is happening within the entire endogenous processes within the system. But in addition to that, you can also see metabolites that are coming from both microbiome contributions, but also lifestyle. So some people will refer to this as more of the exposome. So the influences causing effect on human health, which are really outside of our own genetic makeup. So we certainly know that the influence of a lot of research has been done in the gut bacteria, that bacterial metabolite is actually critical to human health, and if you get dysbiosis of what's going on within your gut, you're going to see perturbations in health. In addition to lifestyle mediators, is this person going toxic on a drug? Are they encountering pesticides in their diet? Are they exercising routinely? These are all effects that can be monitored by looking at the small molecules, which are really not possible when you study more of the other omics. And so for this reason, we like to think of some of the other omics technologies as more of an assessment of the risk of a disease, where studying the small molecules or doing metabolomics is really the actionable information in the current status of the disease. So at the end of the day, as the person in charge, if you will, of being able to detect as many metabolites or as many small molecules as possible in a complex solution, my job is to detect as many small molecules in your traditional biochemical pathway charts as possible, such that when in any particular disease you see changes in specific areas of metabolism, for example, right here, you can identify what is the core biological insight and what is happening in the specific treatment. Now, this example of this slide is actually an individual who has maple syrup urine disease, which is a genetic disorder which is causing a mutation in the enzyme which catalyzes the branched-chain amino acid metabolism. Now, unfortunately, this is a fairly big challenge as an analytical chemist because the breadth and diversity of the compounds in our known endogenous processes is pretty extreme. You have small, really polar molecules like TCA cyclometabolites, lactate, citrate, these types of molecules, all the way up to incredibly hydrophobic, very complex lipids. So you have to have a diversity of methods to be able to detect this breadth of chemical properties. So this is what Metabolon does with our core technology. We actually have 5 different methods that we currently run to be able to capture the breadth of the metabolome. We have what we consider 4 methods that go about to running our Metabolomics platform, or more of a small molecule platform, in addition to an entire platform operator, entire methodology optimized for these really complex, these are the more membrane-bound energy transfer tags, DAGs types lipids. Now, most of what I'm going to be focusing on today is the workflow that we use for our metabolomics data stream, but just be aware that there is an additional stream of data coming from these complex lipids. So the extraction protocol is pretty standard in the field in that we take the plasma sample, or we take whatever the sample is, tissue, could be solid, could be liquid, could be dog food pellets. We've certainly run a lot of those as well. And we do an extraction, and we crash all the macronutrients out, or the macro molecules out, and then what we have left is a supernatant, and that supernatant is what is run on these 4 methods. Now, I'll say that the 4 methods that we run, they're broken into 2 methods that are optimized for more molecules that ionize in positive ion mode, 2 methods which are optimized for negative ionization mode, and then in addition to that, those are subclassified into more polar POS and more hydrophobic POS and more hydrophilic neg and more hydrophobic neg, again, just spanning the scope of what we're able to detect. I'll note at this point that we use all accurate mass Q exactives from Thermo Fisher for the instrumentation here, as well as Waters Acuity UHPLC systems. And we alternate to do between 2 different scan types. We have MS scans and MS scans to be able to collect fragmentation data, and the reason for that will become more clear as we move on. So at the end of the day, it's a fairly simple workflow in terms of collecting the data. Now, that's not to say this is a simple process, and part of what we'll talk about with precision metabolomics is all of the process controls that have to go into place so that you can monitor that these methods are running correctly and that the results that you are getting today are the same as the results that you got last week, is the same as the results you got 2 years ago. And when you start to talk about moving metabolomics into looking at individual samples, this precision and this consistency of data over time becomes critical. So part of precision metabolomics is really the, quote unquote, sort of boring part of running a lab, which is, insane coverage of SOPs, quality control monitoring, as well as internal standards throughout the entire process. And we have certainly honed these methods to drive the precision. And again, I will talk about why that is so important as we go on. But one of the first challenges that traditional metabolomics practitioners really run into is what do you do with the massive amount of data that is being generated from these metabolomics assays. So if you think about a traditional output from one of these assays, you've got chromatographic retention time and the M/Z axis as well, and then obviously intensity. Now, it is often reported in the field, the concept of the number of features that we're able to detect. So in our hands, in a single run of the platform, for a single sample, this is an example of a neg spectrum right here, we are able to detect about 40,000 features or 40,000 pinks in a single sample. So that means when you run all 4 arms of the platform, a single sample will produce anywhere from 160,000 peaks or features up to 200,000 peaks or features. And people love to report these ion feature numbers in the field. And from our perspective, this number is, while not completely meaningless, it is really not relevant to understanding how much breadth of biochemistry you're actually detecting, because you can actually do a lot of things to drive your feature counts up. You can add salt into your chromatographic method and be able to see more features. You can run longer methods and see more features. But at the end of the day, none of that means you're detecting more compounds that are going to be able to actually move your biology forward and your understanding of what is going on in the process. So there's a tremendous amount of redundancy in this data. So we know that a single molecule produces many, many ion features in the forms of adducts, in the forms of multimers, and in the form of in-source fragments, as well as all of the isotopes. Right? So there's a tremendous amount of just redundant data here that is actually getting in the way of cleanly being able to move this forward in terms of making sense of your biological system. So if you think about this plot here where you see a whole bunch of features, and each dot is what we'll consider a feature here. You've got features that are arriving from just artifacts. These are, the phthalates that are coming from all the plastic tubes that everything is stored in, but also all of those redundant ion features coming from the same molecule. In addition to, you just have the metabolites in your sample, as well as some that are statistically significant in your study. But either way, you have many, many hundreds of thousands of these features in a given study. Now, how we handle this complexity of data is to take all of those detected features and immediately search them through our in-house authentic standard library. And I'm going to get into more details about this library in the next slide. But for right now, this library allows us to immediately deconvolute this incredible stream of ion feature data such that we can immediately filter this data and get to just those compounds that are being detected in that sample. And importantly, not only are we taking time to identify the metabolites that are changing, we are also identifying those molecules that are remaining static. So particularly when you talk about looking at, again, N of 1 or individual sample, looking at making assessments on an individual sample, it is sometimes just as critical to understand what is changing as what has maintained completely static. So once we have this list of identified molecules in the sample, only then does that list go into statistics where we can identify those molecules that are upregulated and downregulated, and then you can use that to do interpretation. So this authentic standard library is really pretty pivotal what we're able to do. We call this approach a chemocentric approach. Basically, we are attempting to, as quickly as are possible, remove all of the complications of that incredibly rich data stream that is the feature data stream and move this into a compound-centric approach. And our library is what I call, or what I consider, homegrown, in that it's not a library full of authentic standards that we purchased because maybe somebody said that this could be present or there's the possibility that this potential chemistry can happen. These are compounds that we've seen signals in plasma, we've identified those signatures, we've then confirmed those signatures as this particular entity by way of purchasing these authentic standards. So the library has grown very organically over the years based on us looking at plasma and feces and urine and understanding and identifying the signatures that we were seeing in there. So at this point, our authentic standard library is about 4,000 compounds. We run each of those compounds over each arm of our platform and document the retention time of that molecule on each platform. We document the accurate mass as well as all of those redundant ion features that that single molecule produces in the forms of, as I already mentioned, the in-source fragments and the adducts and all the isotopes. And in addition, we also collect all of the fragmentation spectrum for all of those species. So for that reason, when we detect an experimental signature, we can immediately search it against the library and identify it if it's got a match to the authentic standard library with 3 independent criteria. So an experimentally detected signature has to have the correct retention time to the authentic standard, the correct accurate mass, and the correct fragmentation spectrum to be able to be called a genuine call. Now, a number of years ago, about 10 years ago, a group of metabolomics practitioners came out and put out a publication that I've got listed at the bottom of this slide, where they came up with best practicing for reporting metabolomics data. And one of the aspects that they covered in this publication was how to report the rigor with which an identification was made, or i.e. the confidence with which identifications are being reported. And how they defined, they broke this into a level or a tier-based system. And one of the things that they stated is that for a tier 1 or a level 1 identification was an identified experimentally detected metabolite where you've got multiple orthogonal criteria to an authentic standard match run on the identical platform as that experimental signature was detected on. So that means the vast majority of the identifications that Metabolon makes are tier 1, or in some publications called validated identifications. We do have a few tier 2, and these are clearly labeled in our data such that whoever is looking at this data will have a very clear picture of everything in this category is a validated identification, and these are the ones where we couldn't find an authentic standard to be able to confirm the identity. So this is a very high quality, very high confidence identification. And again, when you think about moving this into precision medicine or looking at individual human health, it is absolutely critical to have high quality and high confidence identifications. If there is any doubt in the identity of the proposed call of the signature, you are not going to be able to move this into human health. The risks are simply too high. So Metabolon has also continuously over the years, in addition to working using this chemocentric workflow, we are constantly trying to improve our process. So I always like to joke around a little bit that back in 2005, we were not doing the greatest work. We were detecting on the order of 100 to maybe 50 to 100 molecules in 05, and we were doing it with a process variability of about 50%. So not great, right? But over the years, as we've made both technology improvements as well as software improvements, many of these big decreases in process variability that you see over the years has absolutely nothing to do with hardware, but actually has to do with quality control steps that we put in place to be able to monitor, for example, how consistently our automated peak integrator and peak detection system was consistently finding peaks and consistently integrating peaks, and just wrapping some quality control around those processes went a long way in reducing our process variability on the back end, such that as of 2016-2017, we're detecting and identifying, importantly, between 2,000 and 2,500 molecules in plasma with an average precision of less than 5%. So it is only at this stage in the game, right, where you're able to detect this breadth of metabolome coverage with this degree of precision and the confidence of the identifications that we are able to make with this library, that we are able to now take this technology into the space of looking at individual samples and assessing individual health. So to just sort of sum up precision metabolomics, precision metabolomics is metabolomics, but it's precision metabolomics on crack, if you will, right, to keep it down low. It is all the boring stuff, SOPs, standardized extraction procedures. All of our staff has to be proficiency trained. The laboratory is actually CAP and CLIA certified, so all of the regulatory things in place that have to be able to support that accreditation are in place. But in addition to that, we have this incredible patented workflow that allows us to do what we need to do quickly and on a time frame that allows us to get data back to a physician who is treating a patient within a time frame that is reasonable, in addition to tackling these big data challenges and all of the redundant measurements of ion features that are getting in the way, such that at the end of the day, we can deliver really high quality data, i.e. comprehensive metabolome coverage up to 2,500 molecules. Again, those are named molecules, identified molecules out of plasma, very high confidence identifications with those tier 1 compound IDs with a precision of less than 5%. And then take all of that incredible data and put it in the hands of a fleet of PhD biochemists who we staff in-house, who can really look at that data and understand what it means when specific branched-chain amino acids are going up, but those fatty acids are going down, and making sense of how that is, what is happening in that particular individual. So now I want to show you some of the, really some of the exciting part of what we do here, and that's just some of the stories, some of the case studies. So we started this work a number of years ago. Baylor College of Medicine actually came to us and said, look, I'm going to give you a challenge. I've got 200 patient samples, completely blinded, and I want you to tell us who's affected, who's diseased, and if diseased, what disease it is. Go. So we ran our precision metabolomics methodology on this sample set, and we were able to absolutely correctly identify all of the unaffected individuals as such, in addition to being able to accurately identify with 100% sensitivity and specificity all of the different diseases that we were encountering. Now, I will say there was one sample that we missed, and we gave ourselves a bit of an out on this one because that particular patient was under active treatment and responding very well to treatment, and so their metabolic signature had come back to normal. We had also, in that same patient sample, were able to see the clinical treatment of the disorder that they were currently under. So again, we were able to see that that patient was being treated and that they were being well controlled in their disease. So it's really very exciting that with this global approach, we're able to do this type of work. Now, really interestingly, if you look at any individual case of these inborn errors, this is a case of arginemia. The clinical measurement for arginemia, or the clinical diagnostic for arginemia, is abundant arginine, right? So you can see in this slide, this picture here, that we had 4 different patients with confirmed arginemia, and you can see that all of their arginine levels are above the cutoff for that definition. But when you look at all the distal markers that we've detected looking at the entire metabolic pathway associated around these diseases, what your eye catches that patient 1 just looks farther deviated out from the rest of the patient samples. And in fact, clinically, that was also relevant because that same patient had also had 3 additional trips to the emergency room because of acute events. So that patient was clearly not being as controlled as well in their disease as the other patient samples. And while you couldn't see this at all within the standard clinical measurement for arginemia, you could certainly see it when you spread out your view and looked at all of the associated global pathways. So by looking at this additional information, you actually get a much stronger sense of the severity of the patient disease. Now, in some sense, looking at metabolic disorders, it is a bit of a low-hanging fruit. The metabolic diseases can cause incredibly large shifts in the metabolome, so they're somewhat easy to catch. We wanted to see how far we could push this technology. So one of the things that we did is we took a patient cohort of self-reported healthy individuals and ran them on the same platform, and just wanted to go in and say, okay, in these self-reported healthy individuals, what are we seeing in these individual samples? So one of the things that we noted is we know that acetaminophen, that there's a certain population of people that cannot tolerate acetaminophen and have liver-induced toxicity as a result of acetaminophen. We had 2 patients in the self-reported healthy cohort that had very high levels of acetaminophen metabolites. Now, interestingly, the patient on the left had higher levels than normal, but nothing too horrible. But when you look at the early markers of liver malfunction or liver disease, these are all starting to get elevated, suggesting this patient is beginning to have some issues with their liver functioning. And this is maybe partially a result of the fact that they have completely low levels of glutathione. So their glutathione is low. They're not able to protect the liver from some of the metabolites that could be causing the damage. Now, interestingly, this other patient, which has had the highest level of acetamin metabolites in their body that we have ever seen clinically in data, but has really no strong indications of liver malfunction, and that's likely because they have a tremendous amount of glutathione, which is this protecting agent within their bodies. Either way, we took this information back to the doctors for these patients, and the doctors immediately said, ooh, we need to get these patients off of Tylenol and start looking at the liver functioning of the patient on the left. So again, this is capable of really understanding, even in these self-reported healthy individuals, it can give you a very in-depth, detailed look at what is going on in these individuals. So what is next for precision metabolomics? Most of our work so far has been validated in plasma, serum, urine, and cerebral spinal fluid in terms of actually looking at these N of 1 or individual samples. But we are also incredibly interested in looking at dried blood spots. Obviously, nobody wants to have to do a venous puncture, particularly within our pediatric samples. And most of our inborn error of metabolism are pediatric patients, infants, in fact. And so nobody wants to have to prick a vein for that. So this heel prick and collection of that blood on a card is the standard of care there. So we are absolutely looking at moving this precision metabolomics into these blood spot cards, of which the real challenge is, and what we are working on actively, is how do you correct for the difference of fluidity, i.e. the hematocrit of the different blood of these patients. So that's something we are really working on. The other thing we are actively working on is moving this and adding metal ion analysis into the workflow. So we know that a lot of these trace metals, these trace nutrients are critical to catalyzing a lot of metabolic reactions. And so we want to be able to see if somebody is depleted in a particular nutrient, that could be the cause of their drama, right? So that is where we are taking the technology. So just in summary, we're able to take this incredible technology, high throughput, industrialized, untargeted metabolomics, to be able to detect a wide range and identify a wide range of molecules in plasma, and to do everything in this process which allows us to then take that technology into the patients, individual patients. So rigorous quality control, patented bioinformatics approach, this chemical library that allows us to make very high quality identifications. So that is all I wanted to talk to you about today. Again, thank you so much for coming today, and I'll let Brandon take it from here.
We'll be answering questions now in the order that they've been received. In the event that we run out of time, we'll answer your question offline as soon as possible.
Okay. So one of the questions that I got is, are you using this technology for, I'm thinking this must be additional, so outside of the N of 1 study. So yeah, absolutely. We are using this technology for all of our metabolomics. So if anybody contracts with us to run their metabolomic samples, the same process is going to be used. This is not just a specific method or a specific set of processes that are used when we're working in individual health. We are certainly also using this for the bulk of our metabolomics practicing. The other question I see here is, what about unknown molecules? So certainly I talked heavily about our known library, but also part of that known library is actually a very large library of what we call unknown or unnamed molecules. So these are molecules that we have seen in data, and we have created library entries for them, so accurate mass, retention time, and fragmentation spectrum, and put them into our library so that we can track them. So in addition to our 4,000 authentic standards, we also actually have a library of 7,000 unknowns that all of our data is searched against. So this can become particularly important when looking at novel biomarkers, or if you're working in a disease where, you're looking for new signatures, that can be very important there. Do you use any GCMS? So we used to have GCMS as a part of the platform. Unfortunately, given our throughput demands, we actually had to retire our GCMS, and we moved the compounds that we detected on our GCMS into other methods. So we are currently not using any GCMS. What I will say is we have some targeted panels within the building that we operate. So for example, we do a GC FAMES analysis for particular clients that that request it. So while we have the technology in the building, it is not used for our global HD4/precision metabolomics workflows anymore. Can you use study samples which are cells and not plasma? Absolutely. We've run 500 different matrices at this point, anything from plasma, urine, feces, cell culture, spent media. I mentioned before dog food kibbles, orange juice, plants of all kinds, plant root, plant seed. You name it, we've run it. We've even done studies in toenails. We've done studies in sweat. So if we can collect it and collect enough material of it, certainly we can run it. Has any metabolomic biomarkers been identified between sleep and physical activity? So absolutely, we actually have some really interesting data. I believe it has either recently been published or might be in process now, where we actually did this incredible longitudinal study of individuals where they had sleep challenges and various physical activity challenges and very dietary challenges, and watched the flux of the metabolites through these different challenges, and you can see incredible changes, certainly with physical activity, and much the responses that you would see as you are in the middle of physical activity. You're converting your tags to free up the fatty acids so that you can utilize them, so you see a decrease in the tag totals in the plasma. We've also done a number of sleep studies where you can start to identify some of the markers of nocturnal behavior and different things related to that. You can also see various stress markers in sleep deprivation studies. Is it possible to distinguish between different oxidation statuses for one molecule? I guess I'm not understanding your different oxidation statuses, so I'm going to put a pass on that one. Can you use frozen samples for testing, or do they need to be freshly obtained samples? So certainly most samples at some point are freshly obtained at some point. We prefer that they're actually immediately frozen for any shipping of those samples to be able to decrease any of the protein functioning in that sample after it has been collected by the person who sampled the data. We have actually done some work looking at a technology where you actually don't even have to thaw the sample to sample it. So there's a technology called cryo-extract where they actually take a core of the frozen sample and core it frozen so that you never have to freeze-thaw your sample. And we're currently working on research within that space to see if there's any benefits to being able to avoid a freeze-thaw sample just prior to our instrument analysis. Do we have public access to the metabolites that are the indicators of different types of diseases? So how we work at this is certainly literature is the basis of a lot of what we do. When we actually go through our interpretation of the data, we'll see markers going up and markers going down, and a lot of times we rely on our own in-house experience with the number of samples that we've analyzed to be able to know that these are the specific metabolic markers of these particular diseases. For example, in the talk I used the example of the markers of liver health. Those are markers that we've seen in every liver tox study that we've run in-house to be good indicators of how well the liver is functioning. But you can also find that information in publications that are out there. So once you start looking for the different indicators of the disease or the different biomarkers of the disease, you are either relying on data that you already have in-house where you've seen it, or certainly the literature precedent for these discoveries.
Thank you for attending our webinar. If you haven't received an answer to your question, we'll get back to you shortly with an answer. In case you'd like to share the replay or listen again, the recording will be available soon on our website at www.metabolon.com.